Hi everyone. Today we're going to study cell transport through two activities. The first activity that we're going to do, we're going to use a model cell to see what can pass across the semi-permeable membrane. Second activity, we are going to look at the solute concentration of different plant matter. Okay, let's get started. The first activity that we're going to look at is we're going to use this tubing here. It's called dialysis tubing, used for dialysis. It is a cellulose membrane. So as a cellulose membrane, it has different properties than a cell membrane. We're going to see what's permeable through this cellulose membrane today. It actually looks at first when you buy it like a piece of tape. Tape. It actually comes in a roll, just like tape. Okay. And what we're going to do, we're going to turn them into cell membrane. And so we open it first of all. It is actually a tube, and we're going to close the other side. Put a knot on the other side. Take a knot. Okay, it's just like tying a balloon. Okay, so you tie it, and we're just trying to seal it up. We're going to put different solutions in this cell, and we're going to see what can pass from the inside to the outside, or what can actually go into the cell as well. So we're going to see how the semi-permeable membrane is actually semi-permeable. So we're going to make a few different solutions today, all 10%. And there's some choice that I'm going to do with some of these solutions. So I'm going to show you everything that I have the ability to make, and then we'll see what we actually make of it. I have, for instance, glucose, like a glucose solution. This is D-glucose or dextrose. I have another sugar too as well. I have the fructose. I have sucrose. I also have some table salt or sodium chloride. And I have albumin. Albumin is egg. And I have gelatin. I also will need a control. I'm going to go make 10% solutions off camera. Then we'll look at what we have before we get started with the experiment. Okay, so I'm in my kitchen and I'm making 10% solutions of sucrose, sodium chloride, fructose, and actually I'm making a 5% of albumin. I don't even know if 5% is going to go in solution. Now, since I'm making percent by mass solutions, the actual number of particles for the solute in each solution is slightly different. So make sure you think about that too. It's important in what's going on here. Okay, normally I'd make molar solutions when I make large quantities, but because I'm making such a small quantity, 10% solutions make much more sense for ease of building and making them. Okay, so I already measured out, for instance, 10 grams of sucrose. To make it a 10% solution, I'm going to add 90 milliliters of water. Now, if I was at school, I would use a graduated cylinder because it's much more accurate than these plastic beakers that I have. But I'm going to use these. The actual percents might vary just a little bit, but they'll be close enough for what we need them to be. Again, 10% solutions of all of them except for albumin, which hopefully will come out to be a 5%. And I'm going to take and add my 90 mils of water to my sucrose solution. Then I'm going to get a clean spoon and I'm going to stir it until it goes into solution, until it dissolves well. Okay, I'm going to do that for the rest of my solutions too. I already measured out masses for them. I will need to make solutions. Also, I'm going to put all these solutions inside of the dialysis tubing. What should be in the control? Come back to that in a little bit. Okay, so I'm going to pour out 90 mils of water for each of the others. For albumin, actually, I'm going to add 95 mils to make a 5% solution. Let's see how well this goes in. With the hair. So, yeah, albumin even 5% isn't going to go into solution very well, but I'll do my best in a little bit to get it better. And you should have guessed that the control, we're going to use water on the inside and outside. So I'm just going to pour in 100 mils of water in here, although the volume doesn't matter really. We'll see in a minute why it doesn't. Okay, those are all what's going to go inside. And then I have containers for what's going to go outside. Now in all the containers, I'm going to put water in them. Okay, and I'm going to put about 140 mils of water in each of them. So now I'm going to have to set up all the dialysis tubes, and so take a break right now, and then I'll get that all ready, and we'll get started. Okay, so I need to grab one of my dialysis tubes that I already have set up, with it open on one side, and I'm going to get a syringe, and I'm going to add 10 milliliters of each solution. By the way, the 10 mil mark is down here, and use the plunger right at that 10 mil mark. I got a little bit of air bubble, so I purposely over pulled some so I can push that air bubble out. A little bit more. Okay, so I'm gonna put that 10 milliliters inside the dialysis tubing. I'm going to close the other side. I'm just going to make sure the ends are dry, and I'm going to get the mass of it. The mass is 11.50 grams. And I'm going to also record the time that I actually put it into the container. So it's going to be 11:29 almost exactly. So I'm going to put that into my control container, and we'll push all the tips in. And we're going to let it sit for half an hour. I'm going to do the next one. I can use the same syringe because that one was just water. Actually, before I do sucrose, I need to make sure that all the others are well dissolved, or as dissolved as I can get them to be. Kind of forgot that step. Salt looks good. Fructose, pretty much already in solution. And albumin, well, albumin's going to be a pain, and I'm not going to worry about it too much. It's pretty good. Okay, so back to sucrose. I'm going to grab a dialysis tube, put a knot on one end, other end. Other end. I need to clean out this container now because I only have one. Yep. And also two left. You can go right out. You can go right out. Okay, I just realized I needed to do cornstarch too, so I'm going to make a cornstarch one. I'm going to make a 5% solution of cornstarch. Everyone else ready outside? Okay, starch is in. Okay, so it's time to start measuring the final masses. So I'm gonna get another paper towel, take out my control, wipe out the ends, and really quickly dry off the outside so there's no water on the outside, and then find the mass. The mass is 11.58 grams. Okay, so did it change any? I'm just going to put that back in there, and I'm going to wait until my next time. Alright, so the next one is sucrose. Pull it out. Same thing. Wipe off the ends. Okay, 12.33. While I'm doing this, actually, what I'm going to do is take samples from the outside of the containers to see what's going on. We're going to do some tests to see if there are substances on the outside. So while I wait for my next time, I'm going to pull out 10 mils of fructose and put that in one of my containers over here. Then I'm going to clean this out. Well, the battery went dead on my camera, but I'm back and I still, at the right time, albumin. This next one. 11.68. Take a quick search and fructose. Okay, while I wait for the starch, I'm going to start doing some chemical tests to see what has moved outside of the dialysis tubing. So we're going to start by testing to see if protein can move outside the dialysis tubing. So albumin is a protein, right? So I have my albumin container here. And I took some of the albumin that went inside and I added it to a test tube. And then I also took water that was originally on the outside and added that to a test tube. Now I'm going to take a sample of albumin from the outside and we're going to put that in this test tube here. Okay, so I have the original water that was outside, I have the original albumin that was inside, and I have what was outside. So we're going to do a Biorets test for this. I think we need to be able to see a little bit more. Okay, I got my goggles on because Biorets actually has some sodium hydroxide in it, and I don't want any of that. And I'm just going to add a whole pipette worth of, or dropper worth of Biorets solution to each. Now the water acts as my negative control here, so I can see what it looks like without anything. And it was what was originally on the outside. The albumin will show me what it should look like with proteins. And I should be able to compare to see what's outside now. I'm actually going to add more because I think my solution's kind of dilute. Okay. By the way, to mix test tubes, usually take your finger and just hit the back of it, and it stirs it up really nicely, creates a vortex. Do that for my outside. Now these colors can be a little faint. I'll go get a white piece of paper to show you what they look like. So I put a white piece of paper, usually helps see the colors a little bit. And you can see that the one with the protein that was added inside is purple. The one that's outside now is still blue like the original solution, showing that protein really didn't transport across. I also asked my children to make me a quick little conductivity tester using snap circuits. So we're going to use this to check to see if salts moved. There are other tests that you can use for salts. You can use silver nitrate to test if there's salt in a solution. So we're going to start with just taking the original salt container. It's the original one. We're going to test to see if there's conductivity. I'm going to put both probes into it, and this is just like the inside of what was inside there. And it should light up. I don't know if you guys can see that. I'll hold it up actually too. You see it's a fun little color changing LED showing that there is conductivity there. Okay, then I want to put this in some pure water and show you that it doesn't light up. Well, hold on, I think I have to rinse off the probes a little bit. Clean water. Let's see if it works. I haven't tried this before until now. Okay, actually I want to show you up close a little bit better. So the LED with the plain tap water is lighting up a little bit, showing there must be some solute in my tap water naturally. Okay, well let's then compare to what was left into our beaker once we had put our dialysis tubing in here. So we're looking at the plastic beaker after the sodium chloride solution inside of the dialysis tubing was in there. And let's see how that compares. Okay, so you can see it's a lot brighter, the LED, hopefully. And that is because there has been some sodium chloride that has exited and passed through the cellulose and gone out into the solution around it. Okay, again, compared to the original salt solution in there, it should be about the same. Okay. It's time for the starch. We're going to test what the starch is on the outside. Under dry paper towel. And we're at two ends. 11.31. 11.31, huh? Alright, anyway, it's okay. They might surprise you a bit. What we're going to do next is we are going to check on a starch test. We're going to see if starch moved to the outside or stayed inside. So, just going to make sure my syringe is clean. Okay, and I'm going to take these here. I'm going to grab some starch from the outside. I'm also going to grab some sucrose from the outside. Okay, I have water from the outside, like we had before. I have the original sucrose that was in the inside of the dialysis tubing, and I have the sucrose afterwards on the outside. Then I have starch on the inside, and then I have the solution on the outside. So what we're going to do is test to see what contains starch. Now the test for starch is an iodine test. Iodine will react with any starch and it will turn it a what's usually described as a purple-black coloration. Grab my piece of white paper too and put it behind so you can see a little bit better. So I'm going to take a pipette full, put it into each one. Alright, so this tests for starch. It does not test for sucrose. So I use the sucrose containers too, just to show you that they wouldn't show a positive test for starch. So a orangish color is a negative result. Just the water shows you that is our negative control again, so it's a negative result. The 2 sucrose is also, they're going to be negative. Okay, and then we check for starch. Okay, so inside with the starch, this is your sort of purple-black coloration that you should get. Outside you can see that it's orangish. So what's that mean? The final test to see what else was transported is we're going to look for simple sugars. Simple sugar test uses what's called a Benedict's test. Benedict's looks for any non-reducing sugars. I'm actually going to switch out containers a little bit here. So for this one, we're actually going to look at the fructoses, the sucroses, and we're just going to check starches too. You'll notice I didn't check all of them for all the tests because I didn't need to waste reagent. I knew they were going to be negative, and so we'll show that in a data table at the end. So to test for sucrose, again I have water, I have the original fructose solution that went in dialysis tubing. I have the solution outside the dialysis tubing, so we're going to see if things got transported out. I have sucrose inside the dialysis tubing, and then what was outside. Then I also have starch originally inside, and then it ran out of room. Starch on the outside. I'll fill that in a second. Okay? Let me do that actually. Okay, so we do the same thing for this test. We add a dropper full. Okay, now there is one difference with this test. Right now you'll see they're all light blue coloration. In order to get the Benedict's test to work, we actually have to heat them up. And so there's a quick way in lab I do it. I just stick them in the microwave for 15 seconds, but I actually don't have a microwave at home. So I am going to put them in a pot and heat them up for a little while, and I'll show you the results in a bit. Okay, my water's heated up and I was about to put them in into the water, but before I do that, I want you to notice that there's already been some color change with a little bit of time. So I'm going to take these all to the pots, put them in for a couple minutes, and then you will see what they look like. Hopefully I don't spill any. Actually, I can kind of show you what's going on. So I just have them in the hot water, and I'm going to let them sit in there for a couple minutes. Actually, I'm going to go put them, rest them on the stove. Okay, so let's take a look at the results. So I'm going to move some of these, try to find the ones I want first. Okay, so this is water. Water is our negative control for Benedict's, and it shows up as a blue color. Okay. Then we had the fructose that was in. The fructose is a monosaccharide. It should show a positive result for a Benedict's test. And so inside the dialysis tubing, there was lots of fructose. Now, so the colorations, guys, usually what happens with the Benedict solution is it changes from a green color to an orange to even a darker color, I think sometimes, if there's lots of sugars. Now, outside the fructose, you'll see it actually started as a slight green color, and as I kept heating it, it got closer and closer to orange. It's not quite the same color but it shows that there is some definite fructose on the outside of the tube. Okay, now for sucrose, sucrose is negative on the inside. It does not, disaccharides like sucrose are considered non-reducing sugars, and they do not test positive for Benedict solutions. Outside also, since it doesn't test positive on the inside, it's not going to test positive on the outside. Starch 2 would not test positive for Benedict solution. You can see my one starch test got a little bit cloudy, the one with the original starch in it. That's only because that starch is going into solution better. On the outside, we already did a starch test for that, so you should kind of understand why the outside looks that way too. So I'm just going to put a light background on here so we can see our results better. And show the camera. Okay, again, really quick, let's take a look at our other results too. These are our iodine test, looking for polysaccharides like starch. And this is our Biorets test, looking for protein. All right, guys. So along with our masses, this is all of our results from this experiment. I'm going to show you quickly too what the other experiment is all about. I am also going to quickly demonstrate part 2. I'm not going to go through every step-by-step part of it. In fact, I'm going to use data that was collected at a different time. But I'm going to show you really quickly how it's done. Okay, the next experiment, I'm going to take some carrots, probably 5 or 6 pieces of carrots, and other plant matter, probably going to use some potato, sweet potato, maybe apples, and I'm going to mass them. Then I'm going to put them into different concentrations of sucrose, different molarity concentrations of sucrose. And I'm going to cover them with a piece of plastic wrap. And I'm going to let them sit overnight. And we're going to look at what happens to the masses of the carrots. If the mass increases, why do you think the mass would increase if it was put in a certain concentration of sucrose? If the mass decreased, why do you think it would decrease? Okay, so we're going to use actually these sucrose concentrations and the change in mass to find the isotonic point of the carrot. That means the point where the solution inside of the carrots has the same solute concentration as what's outside. That can help us figure out actually the concentration of solute inside the carrot. Now, most likely, I'm not going to use a solution with the exact molarity where there's no change in mass. So instead what we're going to do is graph out the data, and we're going to find the point where there is no change in mass. From that, we can then actually, if we know the isotonic point, we can actually calculate the water potential of carrots normally in an isotonic solution. I'm going to show you all the data now, and I want you to analyze it and then make some conclusions. Have a good day, guys.